And I'm really excited to have Professor Carter here. And so Professor Carter is a professor of biochemistry and biophysics at the University of North Carolina. And his work has really centered over the past decades, I guess, on understanding the emergence and evolution of the genetic code. And in doing that, he has been a champion of this rodent-ono hypothesis, this idea that AARS has evolved from complementary strands. And I think maybe is one of the things he's best known for, but he's also been a pioneer in protein deconstruction efforts. So simplifying proteins to understand their evolutionary histories, which is a topic I'm very interested in. And I think he's done a real service for the community because he's articulated a lot of compelling arguments against an RNA world hypothesis, which is a bit of an obsession, I think. So I'm really excited to have him here, and I will pass it off to you, Charlie.
Thank you, Liam. It's a real pleasure to be there or here and to be invited to sort of speculate on topics of my own selection. This is a selection of topics that I've never really talked about, and I don't really often show this ceramic, but it's one of my favorites. It's a gift from a classmate, a college classmate, who got very upset because of our political differences with my my blogging and bloviating in the to our college list. And so he sent it to me just out of the blue. It says, "If I visit reality, it will be as a tourist." And I was so touched by it because it is so, it captures so perfectly who I am, that it's been a treasure ever since. And he has since passed away, but I still treasure this memento of his comment on my commentary. So I'm also thrilled to be here to talk to you about this topic, reciprocally coupled gating, strange loops in genetics, bioenergetics, and catalysis. This is a topic on which I've been inspired over the years by many people. There is a paper that's now available as a preprint. It may be published soon in Biomolecules, and the reference to that is at the bottom. And one of the people that inspired me over the years is Danny Tawfik. And so I want to give a shout out to Danny in the event that he is able to get the recording of this, because many of the ideas, my ideas are influenced significantly by many people, including Danny. So I also want to acknowledge the help of Dick Wolfenden and Peter Wills, who have made very serious contributions to the work that I'm going to talk about. So I've chosen to talk about three different topics, genetics, bioenergetics, and catalysis, because these three areas seem to me to form the foundation of biology. Bioenergetics is the study of maintaining systems away from, far from equilibrium. It's the science of studying the fuels that we use, ATP, how the ATP is converted into mechanical motion, biosynthesis, and control and regulation. Genetics, on the other hand, is a completely different topic that deals with heredity and natural selection. And because of natural selection, it necessarily involves the genotype versus phenotype separation, which is, in my mind, connected very closely with my central interest, which is translation. And finally, I'm going to talk about catalysis. Catalysis is important to biology, not only because it speeds up chemical reactions, but also because it synchronizes. The catalysis that's provided by different enzymes is appropriate to bring all chemistry to about the same rate, despite the fact that the chemical reactions in the cell have uncatalyzed rates that vary by some 20 or more orders of magnitude. So these are all essential components of biology. They're interdependent, as indicated by the black bidirectional arrows. But the most important, most interesting thing to me is that they all are difficult to understand because they each present a different variant of the chicken and egg problem. And the chicken and egg problem is something that I think has been settled in principle by Grasse Tyson, who says in a very concise way that the egg was laid by a bird that was not a chicken. Now, what he's saying, I think, is that there is a historical context, and it's the historical context that is very hard to discover for each of these three areas. So I'm going to start out with an introduction to what I mean by reciprocally coupled gating. And this is, in my mind, a case of what I call process control in biology. And most biologists, most theoretical biologists are very stuck on the idea of feedback. And in turn in something called autocatalysis. And I've indicated a very simple example of autocatalysis here. A molecule of A reacts with a molecule of B to produce two molecules of B. And in this case, the red arrow here indicates that the increase in the concentration of B actually will feed back and accelerate the rate of this reaction in the presence of an excess amount of the molecule A. Now, this isn't a very simple autocatalytic cycle, but Stu Kaufman and Manfred Eigen have both written a lot about how these autocatalytic networks grow. And here's an example of a slightly more complicated autocatalytic network. And if you survey the literature of theoretical biology, autocatalysis will produce a very large variety of these. And one of the problems, and this is a problem that was identified both by Kaufman and by Eigen and by Zathmari, is that when these networks get out of control, they generate side reactions, or which in fact are called parasites, and they become toxic to the survival of the autocatalytic set. And so this is a serious problem. It's a problem that's recognized by these theorists, and they really don't have a really concrete and a robust answer. On the other hand, there is another kind of process control element that I've become aware of in the past year or so, and it's illustrated by this diagram, which is now sort of a logo for this talk. And this involves transitions that connect two different levels of objects or processes or whatever. And they're connected by transitions that in fact are gated by if and only if conditions. So this the transition from level 1 to level 2 happens if and only if something is true. And in return, the transition from level 2 back to level 1 also is gated or filtered or limited, restricted by another if and only if condition. Now, as the talk goes on, we're going to see 4 different kinds of sets of if and only if conditions. And so I'm going to postpone talking about that aspect further. And at this point, point out another aspect of this kind of diagram. You'll notice that the staircases here, in the staircases here, the white steps are the white elements are steps, and both of these are increasing. They're both stepping up, despite the fact that the figure itself is in a plane. This is a characteristic that MC Escher made use of in his diagrams, and is described at great length in Douglas Hofstadter's book, Gödel, Escher, Bach. And the basic notion is that when you have a cycle of this sort that he calls a strange loop, it is characteristic of the Gödel incompleteness theorem, and the incompleteness itself implies a directional force. And I'll work my way through and get back to this idea at the end. Finally, I'd like to mention that that one of the motivations for this immediate work was the fact that Ken Dill, a biophysicist, American biophysicist, well-known American biophysicist, has a paper in press that is that is given to asking the question whether there are actual physical forces that drive the formation of life. And I also will work my way back to that question at the end. So my first example is from bioenergetics, and it involves the reciprocal coupling of ATP hydrolysis and conformational change that enables systems in biology to sustain themselves far from equilibrium. This is a bit of biochemistry that's more specialized for some of the non-scientists here, but I've tried to limit that kind of material from this talk. But I do need to explain that that there is a fuel that all biological systems use, which is ATP. It's a nucleotide triphosphate, which is abbreviated NTP, and NTP hydrolysis drives uphill chemical reactions, and that's how we circumvent the second law of thermodynamics. The reaction of hydrolysis of ATP is one that is explosively irreversible in water, has an equilibrium constant very much greater than 1. It's actually about 10 to the 5th. And that means that the equilibrium is very far over on the side of hydrolysis. However, if this reaction occurs inside a protein, the equilibrium constant, instead of being 10 to the 5, is close to 1. And the reason for that is that the shape of the protein has to change in order to accommodate the chemical change that you see here of converting ATP and water into ADP and phosphate. So this is the group that's split off it's the terminal phosphate of ATP. So in the absence of a catalyst, Frank Westheimer pointed out that the hydrolysis of adenosine triphosphate, other NTPs, is very slow. What this means is that nature has to provide a catalyst in order to accelerate this reaction, and that provision of the catalyst is what enables us to study the conversion of the free energy that's represented by this explosion into conformational changes, which are at the heart of biosynthesis and mechanical work and regulation. So the work that we did on the tryptophanyl-tRNA synthetase led to the identification of what I call an escapement mechanism. And it's this escapement mechanism that I'm going to describe in the next couple of slides. But here you see a very simple example of an escapement mechanism. This is actually a wooden clock. And if I were showing my full PowerPoint, this clock would be actually working. It's an actual video. This is the folio. It's called a folio, and it actually rotates back and forth. And at the far extent of each of the motions, there is a little interaction here that actually slips by. You see one of these pegs slips by a little blade that's kept here, and it slips back and forth so that there actually are two blades that you see here. There's a blade here and a blade here. And this is what enables a clock to convert the rotary motion of the spring or the weight that when you wind a clock, you will either wind up a spring or wind up a weight, and that forces this crown gear, this green crown gear, to rotate. But the escapement mechanism here is what makes it possible for to convert this rotary motion into the precise tick-tock that you see in the clock, because this goes back and forth between alternating the advance of one escapement or the other. And that's what turns the rotary motion into a very precise timekeeping mechanism. This is called an escapement mechanism. Now, it turns out that the tryptophanyl-tRNA synthetase behaves precisely this way. And this diagram illustrates what we've learned about the TRIPRS mechanism. And the first point that I want to make is that that the catalytic step, which is this red, oh boy. Yeah. This red ball here is in fact the magical moment where the reaction, which is shown down here in a cartoon form. This molecule is ATP with its 3 phosphates. This molecule is tryptophan, and in this green conformation, the substrates are brought together, but they're not reacted yet. And at this point, if we look at the conformational free energy, which is this brown dashed curve, this is actually a high energy state. High energy means that it's uncomfortable, it's unhappy, it's unstable. And that also means that if there is a more stable state, which we see here, that's related to this product state, that there is a force that's induced by the difference in free energy from here to here that forces the conformation to change from here to here. And in that process, the chemistry of the active site becomes very precisely organized in such a way that it becomes complementary to the transition state between the reaction, the reactants, and the products. And that transition state is actually somewhere near the midpoint of the chemical reaction. And at that point, the enzyme develops its highest affinity for the ligands. So the affinity for the ligand is given by this green dashed curve. And it occurs at a point where the enzyme is actually moving. In fact, we think it's moving most rapidly. So it's actually something that occurs only transiently in the process of catalysis. Now, I'm going to summarize in each of these slides. I'm summarizing quite a lot of data, and in this case, the data that I'm summarizing on the left is the gate that occurs before catalysis. And this is something that is the result of a combinatorial mutagenesis experiment. This is like a bar diagram of mutations that we made in 4 side chains. These 4 side chains here are about 20 angstroms away from the active site. And the magnesium is the active site metal, which is in the active site. And we actually made all 15 of the mutants of these 4 residues in all possible combination, and that's what this barcode shows. And what we found was that the main effects of these mutations are actually very, very small. Even the 2-way effects are very small. The 3-way effects begin to be bigger, and the 4-way effects are also just a little bit bigger, but this is all very irregular. But nothing approaches the five-way interaction between the four side chains and the active site metal. What that means is that in the transition state, because this is the free energy of activation for the reaction. It's derived from measurements of the rate of the reaction for each of these 15 mutants. We find that the coordinated motion of these five elements, the four side chains removed from the active site and the active site itself, actually accelerate the rate by 100,000-fold. And that means that because these four side chains all have to move together, that means that the enzyme has to change conformation in order to form the conformation of the enzyme that's complementary to the substrate. Now, that means that catalysis occurs if and only if there is a conformational change. On the other hand, the reaction is also gated from the right. And the gating in this case is indicated by these free energy surfaces. So there's the pre-transition state product, which is this green state, the product's conformation, which is this magenta state. And at the top of this roller coaster, with the pyrophosphate bound, the energy difference between here and here is not favorable, but it's unfavorable to go to the product form. However, if we remove the pyrophosphate, then this reaction becomes favored. So this downward turn of the conformational free energy occurs only if the pyrophosphate is no longer there. Now, this leads to a paradox, and the paradox is that the gating from the right-hand side says that the catalysis doesn't occur unless, it says the conformational change doesn't occur unless the pyrophosphate is released. Whereas from the left-hand side, it says that the catalysis doesn't occur unless the conformation changes. So here we have a real a conundrum, a puzzle. This puzzle is actually relevant to all of the ATP consuming reactions in the cell. And for that reason, I believe that, that this kind of analysis can and should be done to all transducing enzymes. So this brings us to our first strange loop, the reciprocal coupled gating that efficiently converts the delta G for hydrolysis, ATP hydrolysis, into biosynthesis. And it's summarized here. Here's the gate that I showed you, the escapement mechanism, and the escapement mechanism is administered, these two blades. The first blade is administered by this if and only if condition, that the transition state binding can occur only if the conformation changes. And this blade tells us that the conformation can change only if catalysis occurs. That's the paradox. And that's why this is called a strange loop. This paper, this is work that's described in the reference that's given here. So my second strange loop concerns genetics. And just in a nutshell, what I'm going to try to tell you is that protein folding rules and genetic coding rules are reciprocally coupled filters that produce functional aminoacyl-tRNA synthetases and genetic coding. So I've summarized the various theories of the origin of life. I'm very interested in, and I think that the origin of life is the driving force in my work. Theories of the origin of life basically fall into three categories depending on what you think came first. Whether you thought metabolism came first, or replicators, or cells. And there is a problem with all of these theories, which is represented by this question mark, and that is that there's no way to get from these very basic prebiotic systems to a living thing. And I've represented life here by the last universal common ancestor, Luca. So this question mark is actually filled by the aminoacyl-tRNA synthetases. And I've got to unwrap this diagram a little bit for you. The machinery that nucleic acids are polynucleotides, and they contain the information in genes. But that information doesn't do anything in life to speak of unless it's interpreted by the process of translation. So translation is the process of making proteins. Now this is a protein, for example, and this is a protein. This is a class 1 aminoacyl-tRNA synthetase. This is a class 2 aminoacyl-tRNA synthetase. And the relationship between the information in genes and the information in proteins is that in nucleic acids, the bases in the nucleic acid are chunked in groups of 3 that are called codons, and there is a mapping called the genetic coding table that defines what amino acid belongs to each codon. And this is the machinery. This set of aminoacyl-tRNA synthetases and their cognate tRNAs are the machinery that is responsible for translating the genetic code. Now, these molecules, the synthetase and its tRNA, bind very closely, tightly to each other, and also very specifically to each other. And that's why we call these cognate pairs. So the last thing I need to tell you before I proceed is that that one of the strangest things about the translation machinery is that the aminoacyl-tRNA synthetases actually come in 2 flavors, class 1 and class 2. And that's what I'm going to talk about on the next slide. I'm first of all going to tell you how we got into studying the aminoacyl-tRNA synthetases. So if you superimpose the structures of the enzymes from one class, and I've done 4 class 1 synthetases here and 4 class 2 synthetases here, a very remarkable thing emerges, which is that in the vicinity of the active site, these enzymes all have exactly the same structure, both the class 1 and the class 2. And the class 2 enzymes are not at all the same as the class 1 enzymes. On the other hand, elsewhere, there's a lot of foliage here that is not the same. So I've sort of introduced a metaphor to deal with this. The catalytic region here, which we've called this catalytic region an Urzyme after the German prefix Ur, which means fundamental or primordial or authentic. And we believe that the Urzymes are in fact the chassis and drivetrain of a car, and that the rest of this material here is the make and the model. And this metaphor is actually quite detailed and quantitative, but I won't go into that at this point. The second point about the 2 classes of synthetases is this hypothesis that Liam told you about, which is the Rodin and Ono hypothesis, which is that the 2 flavors of aminoacyl-tRNA synthetases come from opposite strands of the same ancestral gene. This slide is a compilation of a very large amount of work in which we actually have, I think, established sufficient evidence that this hypothesis now has to be considered to be essentially proven. I won't go into that, but I think there's very convincing evidence that this hypothesis is true. And the most important data is that we've actually made a molecule like this, which is a gene that actually encodes this protozyme here, which we call Adam in Greek to avoid detection by the Creation Society in the United States. And this is a class 2 protozyme, which we call Eve. These protozymes are actually small fractions. They're about half the size of the Urzyme. And we were able to actually synthesize this gene because the antisense alignment of the two strands is so obvious for the protozymes that it was basically straightforward to take Rosetta and design the base sequences for this gene, and then to insert this gene in opposite directions with protein expression vectors, and then we could characterize the chemical activity. And it turns out that they both accelerate amino acid activation by a millionfold. So this idea that Rodin and Ono have proposed is an unspeakably bizarre idea. It's really quite jolting, because biologists consider the nucleic acid of a gene to consist of a coding strand and a template strand. The template strand is there in this context only to provide the instructions for copying the coding strand. But in the case of the Rodin-Ono gene, or the protozyme gene that we've made and tested, both strands act as both coding and template. And it's able to do that because each strand has a different base sequence, a complementary base sequence with no distinct information, no unique information in the opposite strand, but the opposite strands have different interpretations. And that's a molecular implementation of this diagram here, which is either a forest or a tiger, depending on which, whether you look at the figure or the ground. So the figure of this figure are the trees, the ground is the tiger, and the figure and the ground correspond to the two strands of the Rodin and Ono gene. This is a profound unification in biology because the descendants of the class 2 synthetases and the descendants of the class 1 synthetases, I believe, can be found widely distributed in nature. One of the descendants of this protozyme is probably myosin. Another descendant of the class 2 family is actin. So the second element of the strange loop in genetics involves the way that information is stored in transfer RNA. So transfer RNA is this molecule here. I've highlighted the bases in this part of the molecule, which is the acceptor stem. The amino acid actually gets attached up here. The red bases down here are the anticodon. This is what actually is able to read the sequence of codons in a gene and position the amino acid that's bound up here in line to be assembled into proteins. Now, the magic that Dick Wolfenden and I have discovered is that the information about protein folding, which is represented here, is actually embedded into the structure of the tRNA. And so I want to explain what this diagram is. It's a thermodynamic cycle of phase transfer free energies, and these Ks are the equilibrium constants for the reactions that are given here, and the reactions themselves are distributions of side chains, amino acid side chains. Every one of the 20 amino acids has a side chain that's different, and what Wolfenden did was to take those chemicals that were exactly the same things as the side chains, which do exist separately. And then he measured the distribution between vapor and cyclohexane and between water and cyclohexane. And this experiment is very much like taking a salad dressing, a mixture of salad dressing, and shaking it up and to equilibrate all of the components, and then setting it on the shelf so that the phases separate. Some of the amino acids will go to the water phase, and some of them will go to the oily phase. And cyclohexane here is something that represents the oily phase. So the first thing that Dick and I did was to show that these two equilibrium constants actually explain the rules of protein folding. We found that we could get a very high correlation between the appearance of side chains on the surface of proteins and their distribution coefficients between water and vapor. Between water and cyclohexane. So the next thing that we learned, was that the bases in tRNA, which are represented here, are highly correlated with these two equilibrium constants. The equilibrium constants for vapor to cyclohexane are all found here, and the equilibrium constants are all represented by bases here. And these equilibrium constants are represented by bases in the anticodon. Now, the last thing I need to tell you is that the tRNAs actually recognize, the tRNA synthetases actually recognize opposite grooves of the tRNA acceptor stem. So this is the class 1 synthetase, and this is the class 2 synthetase. And the identification of the operational code in the bases in the acceptor stem enabled us to work out what the structural features are of the aminoacyl-tRNA synthetases that allow this kind of specific recognition of this region of the molecule, which is sufficient to begin to implement a proper genetic coding relationship. So there's a sort of a three-way intersection of the physical chemistry of the amino acids, which is here, the specific bases that dictate recognition by the tRNA synthetases of each class and for each kind of amino acid, and the genetic coding table. Now, the last thing that I need to tell you about the synthetases is this very curious factoid. So these two cartoons represent the class 1 and the class 2 tRNA synthetases, TRPRS and HisRS, and these are the Urzymes from those two synthetases. And if you look at the active sites, which are represented by these balls, these are the side chains that interact with the substrates. Here's the substrate here. This is the tryptophan, and this is the ATP. And these side chains are all highly conserved in class 1. And the remarkable thing is that every one of these requires the class 2 active site to activate it and to acylate its tRNA. And conversely, the active site of the class 2 synthetases is defined by highly conserved residues that are all blue here. That means that they all are activated by this active site. So what that means is that you can't construct this active site without this Urzyme, and you can't construct this Urzyme without this active site. So the two flavors must have emerged at the same time. And they are interdependent in an extremely important way, which is diagrammed here. We have our rhodine and ono gene, and we have the class 1 synthetase and the class 2 synthetase, and the active sites here are all derived from amino acids that are activated by this enzyme, and the active sites here are all activated by this enzyme. This is a very strict interdependence, which means that it's hypercycle-like, and it helps assure survival. So this slide is a bit complicated, and I have to take it in baby steps. This is a picture of what I call sequence space. Sequence space is the very large space of all of the protein sequences, all of the possible protein sequences. Now, there's a very small number of them here that represent the class 1 and the class 2 protozymes and urzymes. And the fact that they come from opposite strands of the same gene means that they populate different regions of sequence space by definition. Just you get 2 for the price of 1, as I said before. So there's the rhodine ono gene that gives rise to translation products, which are these noodles here. And then we introduce the protein folding rules, which as these guys pass through the protein folding rules, they fold. They assume structures, and those structures themselves can in turn enforce the rules by which they were made. This is, it doesn't take, I think even a non-scientist should be able to understand or appreciate at least the majesty of this conundrum, that the tRNA synthetases are examples, they're unique examples of proteins that enforce the rules for making proteins. So that brings us to the next strange loop. If we subdivide this class 1 into class 1A and class 2, then we have 4 coding letters. And so this is actually also a model for the expansion of the genetic code that I'll get to later in the talk. So here's our second strange loop. The aminoacyl-tRNA synthetases must enforce the rules by which they are assembled, and they do so by combining the protein folding rules of amino acid chemistry and the genetic coding rules. So the enzymes fold and are active only. They're active only if they fold, and then they fold only if they obey the coding rules. So this is this has exactly the same formal structure as I described before for the escapement mechanism. So I'm going to turn now to a brief discussion of catalysis, because it turns out that catalysis also exhibits this strange loop phenomenon. And the example that I'm going to tell you about is that rate accelerations by molten globules represent the simultaneous formation of 2 extremely rare species, the chemical transition state and the complementary enzyme configuration. So in order to do justice to this, I have to unwrap this for you as well. There are 3 different states that proteins exist in. The folded state is called the native state, and it's represented here by a set of secondary structures. These are alpha helices, and they're held together in a very precise way by these side chains, which are the gray rectangles. And if we heat this structure up, it forms an intermediate state. Well, if we heat it up far enough, then it becomes totally denatured. So this is the unfolded state, in which case all the side chains and all the secondary structures are broken. They're basically denatured. But there's a very interesting intermediate state in which the secondary structures remain, but the side chains lose contact with each other and form a fluid kind of structure. And that's why it's called a molten globule. So it's molten globules that I'm really interested in here. And the reason that I'm interested in it is because we showed we have now shown that 2 of the AARS Urzymes are actually molten globules. Now, this is a very complicated slide. I'm not going to try to tell you all about it, but the most important thing are these arrows. So the arrows refer to the x-axis here, and you've got to look at the numbers to see that this arrow here, this bidirectional arrow, is 0.8 of a parts per million, and this one is 0.7 of a parts per million. This is the LURS Urzyme. So the consensus interpretation of this is that this is a diagnostic for a molten globule. So the next thing I want to show you is that this we are sort of late to the party on this one, because Don Hilvert actually did a beautiful experiment. He did this experiment in which he first of all characterized an Urzyme form of the enzyme chorismate mutase, and you can see that that has a dispersion here of 0.5 a part per million, which is along the same order of magnitude as the 0.8 of a parts per million for the 2 Urzymes. But if you take this material and you add to it a transition state analog inhibitor. Now, what is a transition state analog inhibitor? A transition state analog inhibitor is a stable molecule that looks for all the world like the transition state between the substrate and the product of the reaction. So it's what the enzyme is actually looking for in order to catalyze the reaction. So if you add the transition state analog inhibitor, you suddenly get this spectrum. Sorry, I should have said that these are NMR spectra that are basically HSQC spectra. They're rather simple to measure, and they're very highly diagnostic of the structural difference between a folded protein and a molten globule. So molten globules are very fluid. They're continually trying and failing to fold. The experimental diagnostic is the poorly dispersed HSQC spectrum. And if you feed the molten globule something that looks closely like a transition state for the reaction it's supposed to catalyze, it causes it to fold. Now, the other thing I was going to tell you about this is that there are 2 other diagrams from this paper in which they do the same experiment with the stable native enzyme. And what they find is that even in the absence of the transition state analog, the native enzyme spectrum looks exactly like this. So in fact, the only distinction between the molten globule and the native enzyme is that it's just not properly folded. It has the same catalytic activity. In the case of chorismate mutase, it has the same catalytic activity as the native enzyme, but it has a totally different structure, which is molten. So that leads us to a conundrum here that is very aptly formed by this strange loop. The transition state forms if and only if the active site is of a molten globule is present, and the active site is present if and only if the transition state is present. So here again, we have a very similar paradox, which is very difficult to resolve unless you realize that it's part of this strange loop. And what it tells us is that there is something unknown as yet and quite magical in molten globular proteins that allows them to form very tight binding to transition states of reaction, despite the fact that in the absence of the transition state, they have almost they're totally molten globular. Now, there are some numbers that we can associate with this to sort of dramatize why this is so relevant. And the transition state itself is present at about 1 in 10 to the 15 molecules of substrate, and the active site is present at a comparably small fraction. Now, when you combine a substrate with a folded enzyme, there's no problem because the enzyme has the active site there all the time. But in the case of the molten globule, that active site isn't there unless the transition state forms, and the transition state and the active site together have a joint probability of something like 10 to the minus 30, and yet catalysis occurs. It's a profoundly interesting and as yet unplumbed problem. So I'm going to finish up with a short section on optimizing the coding alphabet and a guide and guiding the evolution of the proteome. So there are three constraints on the fidelity of tRNA aminoacylation by aminoacyl-tRNA synthetases. I've diagrammed them here as a triangle. The first one is thermodynamic. So several amino acids resemble one another much too closely to differentiate them by a single binding interaction. And this is an unalterable thermodynamic limit. The alphabet size, on the other hand, describes or defines the kind of nanoscale engineering that you can do with a limited number of amino acids. So if you improve the number of amino acids that you can code, so that's the number of coding letters, then you can improve the nanoscale engineering and you can improve the specificity of the interaction. And finally, there's a very interesting new phenomenon that's described in this paper by Peter Wills and I, in which Peter actually proved that that the reservoir of isoaccepting tRNAs is a buffer against misacylation. And the way that works is this: if you have a binary alphabet, then because there are 64, roughly 60 codons, because there are several stop codons, if you have a binary alphabet, then every codon is assigned to one of the or the other of the two binary letters, and that means that you have a redundancy of about 30 codons for each of the two letters. Now, as you increase the size of the coding alphabet, that redundancy goes down. And it turns out that, that these isoaccepting tRNAs help to ensure minimal information loss as the alphabet increases from 2 to 20 letters. So it turns out that, and I'm not going to show you all of these, each of these pairs of influences on genetic, the specificity of aminoacylation can be described as a strange loop. I'm only going to be talking about the alphabet dimension, and that's shown on the next slide. So the basic part of this slide are the phylogenetic trees of the 20 aminoacyl-tRNA synthetases. These are the class 1, these are the class 2. This is an entirely hypothetical tree. There's almost no evidence for it because as yet we don't know what these trees look like. Nevertheless, the diagram is useful because it allows us to identify these points at which the coding alphabet has to decrease. So if we go backwards in time, we've got 20 synthetases here. And if we go backwards in time, there are points at which two synthetases for similar amino acids become a single synthetase. Here and here and here and here. So we go from 20 amino acids to 16 amino acids, and from 16 amino acids to 8 amino acids, and so forth. Now, Anton Petrov in Lauren Williams's lab has published a very interesting paper in which he speculates, or the authors speculate that the ribosomal proteins actually represent snapshots frozen in history of what we see down here. the evolution from secondary structure to molten globules to tertiary structure. I've already told you about molten globules, and I've already told you that two of the Urzymes that we've prepared, we've shown are in fact molten globules. So a binary alphabet with just two letters, two types of letters, A and B, we call them small and large because that's what the data seem to show. We believe are sufficient to enable nature to form sequences of a rhodine and ono gene for protozymes. Because the only thing present in protozymes is secondary structures. So really all we need to do is define these two kinds of patterns, A, B, A, B, and AA, B, 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 AA. So in time, the alphabet gets bigger, and we have 4 letters to deal with, and that we think may enable us to get molten globules, and similarly we might be able to get tertiary structure with 8 and more coding letters. So these are speculative ideas, but they are supported by the kind of bioinformatics work that Anton has published, and also by work that Peter and I are doing. Now, Peter has developed software now to recognize these points as we reconstruct the phylogenetic tree. And that is going to, we hope, materially transform the analysis of these phylogenetic trees. There's another paper that we have that's now in press that describes a preliminary use of phylogenetic methods in the class 1 family. So this brings us to the final example of a strange loop in which the coding precision is related to the alphabet size is alphabet size and the coding and the synthetase precision are related by this strange loop. The alphabet size, the precision increases if and only if the alphabet increases, and the alphabet size increases if and only if the tRNA precision increases. So I'm going to now finish with one slide in which I try to pull all of this together. And I'm very interested to have feedback and as to whether this makes sense. What I've described for you is formally presented in this scheme. As I told you, there are 2 XNOR gates. Now, I guess I didn't talk about XNOR gates. I called them if and only if conditions. But in fact, in the language of computer science, an if and only if gate is an XNOR gate. So the XNOR gates are defined by antecedents, which are the first level, and consequence, which are the second level. And in the case of a strange loop, the strange loops that I've described that keep popping up all over biology, 2 of these XNOR gates are linked tip to tail by these curved arrows here, in which the consequent of the first is actually used as the antecedent for the second. All right, now that creates a 2-dimensional force diagram, and that's what I'm going to try to convince you of. The first force is in the vertical direction, and it involves the filters that are provided by these XNOR gates. And I suggested to you earlier that the XNOR gates are actually filters. That's another word for gate. They're filters that actually eliminate parasites, and these parasites are these side reactions that don't obey the XNOR condition. And so the XNOR gate is a filter, and the filter is related to survival. And what it does is to limit the flow of material or processes through this pair of reciprocally coupled gates. And my suggestion is that that is very analogous to a gravitational force in physics. A force is something that induces a change in the direction or motion, the acceleration of some motion. And gravity is the classic example of that. And we find that the elimination of parasites is in fact I'm suggesting that that elimination is in fact a force that acts like gravity to make sure that what goes through this cycle is only the stuff that we're really interested in. So in the second direction, in the horizontal direction, we have incompleteness. And incompleteness is one of these very interesting ideas that Gödel introduced, and which Doug Hofstadter explained in his book, Gödel, Escher, Bach. And it's enabled by the tip-to-tail coupling of these two XNOR gates. That's what creates the strange loop. And the idea that I want to leave you with here is that incompleteness is, most of the people that talk about incompleteness think of it as the cup half empty, because it means that you can't have consistency in a logical system. But when you think about molecular biology, incompleteness is actually a cup half full, because it's a reservoir of opportunity, and that is the substrate for discovery. So the last thing I have to say, I guess, is that is to remind you that I think that these two effects are in fact biological forces, and that they are analogous to gravity and to the chemical potential of what happens between partition that has a high concentration of a particular molecule and another that has a low concentration. There is a force that's generated that leads to the transfer of molecules out of the high concentration region into the low concentration region. So I'm going to leave you with that, and hope there's still time for questions. I hope I haven't exhausted all of your patience. Thank you very much.